Right, welcome to the revision video for Module 7, Infectious Diseases. I'm just going to go through all of the syllabus dot points in this video, and it will be split into two parts because this module is quite a long one. So we'll split it into infectious diseases. We'll go through the introduction to the pathogens and what they cause, and then in the second video, we will go through the responses to those pathogens. So looking at the human immune system and looking at some of the prevention methods and treatment methods to infectious diseases. So just a little recap, what is the content focus of this module? It is tied really nicely to Module 8. So Module 5 and 6, they are linked together, but Module 7 and 8, they have a common kind of topic of discussion, which is diseases. Module 7 is all about infectious diseases. Module 8 is all about non-infectious diseases. So definitely they are linked together. You will see questions that combine both of them, but they are talking about different things. So how I've structured this PowerPoint is we'll go through each of the inquiry questions, make sure that you can answer all of them, because especially for trial exams, teachers really like testing these inquiry questions and syllabus dot points. Make sure you've read through all of them and you can write things about each of these syllabus dot points. But this PowerPoint here really nicely summarizes everything. So if you are cramming for your final exams, which I hope you are not, you can just go through this PowerPoint. All right, first of all, inquiry question 1 delves into the pathogens. We look at a small experiment, which is microbial testing, which you should have done at school. Robert Koch and Louis Pasteur, and also some diseases in agriculture. You are required to remember one animal and one plant disease, so definitely commit that to memory. First of all, pathogens. You have to know how to differentiate the different types of pathogens. Okay, virus, prions, fungi, protozoas, and macroparasites are the pathogens that I do want you to remember. One thing that I want you to know is not every microbe is a pathogen, and some microbes can become pathogens later on in their life cycle. So, for example, the gut microbiome, we have a lot of good bacteria in our gut. However, they can become pathogenic if there is an imbalance in that gut microbiome. So, just because something is called a microbe and not a pathogen yet, later on in their life, they can become pathogenic. Because what is the meaning of a pathogen? A pathogen is anything that is disease causing. Okay, so they are a disease causing organism or microbe. They can be microscopic, so they are known as microbes, or they can be macroscopic, so they are known as macroparasites. So thinking about tapeworms, you can see them with your naked eye, which means they are a macroparasite. Microbes, for example, viruses like COVID-19, you cannot see them with your naked eye. You need a light microscope, or for viruses, probably an electron microscope. You call them microbes, and they are pathogens. Okay, so pathogens, they can be spread via different ways. Physical contact, contact that is not physical. So sneezing, for example, into the air and someone breathes it in. This is not direct contact. It can also be spread via vectors. Okay, so thinking of malaria, malaria is carried by a mosquito vector. And that mosquito just carries the pathogen and transmits it to the host, which it can infect. And I really want to again emphasize you'd need to know each of the characteristics of the different types of pathogens. Right, so the first one I'm going to go through is prions or prions, whatever you like to call it. And this list here goes from smallest to largest. Okay, so remember the order of size as well, because you can get a multiple choice question that asks you what is the smallest out of these pathogens, and you will have to circle prions. Okay, so prions, what are they? They are just proteins. We all know what proteins are from Module 5, a chain of amino acids. Before prions, they are proteins that are misfolded. Okay, so they are non-working prions. They are disease-causing proteins. And what they do is they cause other proteins to also misfold. Okay, so not only are they not working, they will eventually spread to other working proteins, and then they will cause those proteins to be abnormally structured. These eventually will build up, and this leads to tissue damage and death. Because remember, the function of proteins, they do everything in our body, such as enzymes. Okay, enzymes speed up reactions, chemical reactions in our body. Without those, we will not be able to function basically. So prions, they are really bad because they cause the rest of our proteins in our body to misfold. An example of that is mad cow disease. Mad cow disease was a really big problem back in the days and especially in Europe, affecting cows, and it caused them to act really weirdly, hence the name mad cow disease. And very luckily, Australia didn't get any cases of mad cow disease, which is why Australian beef is so highly renowned in other countries like Europe. Cure is also another one. And for each of these pathogens, I want you to remember a specific disease that is associated with them. So I want you to be able to provide me an example of a disease that that virus or bacteria causes. Okay, because a potential question that you can get in the exam is name a virus, describe its structures, and then what disease may it cause. So definitely remember at least one disease for each of these different types of pathogens. Right, the next smallest is viruses. What are viruses? Viruses are just DNA or RNA, so nucleic acid, inside a protein coat, or also known as a capsid. Okay, so viruses, they are just these nucleic acids as shown here, and then a protein coat around it. You might see this characteristic kind of structure here that looks like this. This is known as a bacteriophage, and this is a particular virus that affects bacteria. Okay, so if you ever see a diagram that looks like this, I'm sure you have in our lessons as well, then this is a bacteriophage. What is an example of a virus? I want you to reference COVID-19, okay? Because you know about COVID-19, you went through the pandemic, you really don't have to remember any other example if you know COVID-19. Okay, so the SARS-CoV-2 virus causes COVID-19, which is a respiratory disease. Moving on to bacteria, this is the third smallest pathogen. Bacteria are unicellular prokaryotes. Okay, this should be in your brain by now. We learned it from year 9 and 10. So unicellular prokaryotes. What does prokaryotes mean? It means that they don't have membrane-bound organelles. Unlike eukaryotes, which is us, you and me, okay, eukaryotes have membrane-bound organelles, which means we have that membrane around all of our organelles, like our nucleus. It has a nuclear membrane. Prokaryotes don't have that. Okay, and they cause diseases like salmonella. Salmonella is commonly known as food poisoning. It is found in our chicken and eggs if you don't cook them properly, and does cause a lot of symptoms like diarrhea and vomiting. All right, next we have fungi, and they are eukaryotic, which means they do have membrane-bound organelles. They can be unicellular or multicellular. Okay, so they can consist of one cell or many cells. How do we differentiate between fungi and other plants? Okay, because they are quite similar, is they have cell walls made up of chitin instead of cellulose. Okay, so for plants, they have cellulose walls. Fungi, they have chitin walls. Some diseases include ringworm and thrush. Okay, very common skin conditions. An athlete's foot is another one. Okay, fungi thrive in wet, moist environments, which is why we call that athlete's foot, because athlete's foot is a fungal disease that prominently exists between toes of athletes. Why? Because athletes do get sweaty when they run or do like marathons, and fungi love these sweaty, warm environments to live in. So that's why it's known as athlete's foot. A very good example to remember there. All right, protozoas is one that is commonly forgotten. Okay, we don't hear a lot about protozoas. It doesn't cause too many diseases. The common one is malaria. A common example of protozoas is amoebas. If you've watched Amoeba Sisters videos, they are protozoas. And protozoas are eukaryotic unicellular organisms that lack cell walls. So they don't have cell walls at all in comparison to fungi, which have cell walls of chitin. Okay, a disease includes malaria. Okay, malaria is a disease that is caused by mosquitoes and transmitted via mosquitoes. And it is a very dangerous disease that is not in Australia, which is very good for us. But a lot of developing countries have that in climates that are warm. Moving on, macroparasites. We've mentioned this before. It is a parasite that can be seen with the naked eye. They include tapeworms and guinea worms. Okay, so anything that you can see with the naked eye, they're known as macroparasites. All right, the next syllabus stop point looks at pathogen adaptations. I want you to remember a few, especially for viruses and bacteria. Those two are commonly asked. The rest of them, have a look at this slide and try to remember them. Viruses, the very common one that we look at is high mutation rates. They can have antigenic drift or antigenic shift. Go back to our booklets to have a recap of what they mean. But what this causes is just every year we have people who get infected with the yearly flu, which is caused by a virus, and some people do get resistance against them, okay, because they have developed immunity against them. But viruses, they want to live, they want to be able to infect as many people as possible. So if they infected that person last year, then this year they cannot affect them anymore unless they have mutated. And once they mutate, now they are known as a different strain. And the immune system, even though they have previously been infected by this virus, they are no longer immune to this new virus. Okay, so the high mutation rate just evades our immune cells and therefore they get infected again. Another thing is vaccines. Okay, vaccines target specific strains of viruses, but if they mutate very quickly, then viruses can no longer be effective to those new strains. For example, COVID-19, right? There was so many different types of mutations of COVID-19. We had like the alpha strain, the beta strain, the gamma strain, delta strain. I'm pretty sure they're up to like epsilon or something. But vaccines are not effective against the newer strains. The vaccines have to be redeveloped every year to target those new strains. So that is an adaptation that viruses have, high mutation rates. Another adaptation that they have is that they can live for a long time suspended in the air. Remember how viruses reproduce? They have to invade a host cell and they inject their RNA or DNA into our DNA and they use our cells to kind of replicate. So they're kind of non-living cells, and they can live in the air for a long time. And what that does is just allows more people to breathe in those viral particles, and therefore more people get infected. Read through bacterial adaptations as well. The common one that students remember is they have these things known as fimbriae. Okay, they are just long sticky threads that come out from the bacteria. What that does is it allows them to adhere to the urinary tract. Okay, so UTIs, when you get UTIs, very common in females, they are caused by E. coli. Okay, and E. coli sticks into the urinary tract and it prevents the urine from expelling it. So you cannot actually naturally get rid of urinary tract infections unless you take antibiotics because the E. coli just sticks in your urinary tract for a very long time because of these fimbriae here. Have a look at the rest of these. I do want you to kind of remember some other bacterial adaptation, pathogen adaptations as well. All right, moving on. We need to know how a pathogen can be transmitted. Okay, and this is explained by the chain of infection. Okay, you will see this in exams. Okay, so I'm going to highlight this here. Be able to kind of explain each part of the chain of infection. First of all, the infectious agent is needed to start off the chain of infection, right? We need the pathogen in order to have an infectious cycle. All right, the pathogen then needs to have a reservoir. Okay, a reservoir is where the pathogen can grow, where it can live and reproduce. This reservoir can be someone who is infected, or it can be like a vector. Okay, vectors often are reservoirs for where the pathogen can live and grow and peacefully reproduce. Next, I need a portal of exit. So let's kind of think of this as like someone who is infected with COVID-19. Okay, we'll put at the start of the cycle, that's the SARS-CoV-2 virus. So SARS-CoV-2 virus, that is the infectious agent. And we're going to call the reservoir as someone who has already been infected with the SARS-CoV-2 virus. They have an active virus in them. They are known as the reservoir. Okay, so someone has been infected with COVID-19. COVID-19 is now reproducing inside of them, causing symptoms, but COVID-19 is living and growing in them. Okay, the SARS-CoV-2 virus. After the virus decides, okay, I've reproduced enough inside this person, I want to infect another person now, they need a portal of exit. This can be through digestive systems, so through feces, Okay, it can be through vomiting, not really a symptom of COVID-19, but more for gastro diseases. You do get vomiting and diarrhea. But mainly for respiratory illnesses like COVID-19, we have coughing and sneezing and all of those symptoms, runny noses, that just allows the virus to escape from the body. Okay, next we have a mode of transmission, so direct contact, indirect contact. Direct contact is just looking at physical contact between an infected individual and a susceptible person. So there has to be physical contact that could be through hugging, through kissing, through sharing of saliva, through sexual intercourse. That is all direct contact. Indirect contact when there is no physical contact. Okay, pretty simple definitions here. That can be through airborne transmission, contaminated objects. So if you touch a door handle and then someone else touches that door handle and rubs their face, they could be infected by that virus. And from animal to person. Okay, vector transmission is a type of indirect contact. One thing that I want to be very particular about is sneezing. Okay, sneezing can be a form of both direct and indirect contact. So if a person sneezes directly into your face and you breathe in those spit particles or it lands on your tongue, then that is direct contact. Okay, but if the person sneezes into the air, the viral particles remain suspended in the air, and then later on, let's say 30 minutes later, someone comes and breathes in those air particles. That is known as indirect contact. Because was there contact between the infected and the non-infected person? No, right? It was only the air particles. So that is known as indirect contact. All right, so that's the mode of transmission. Next, we need a portal of entry. Okay, it needs to enter the new host somehow. A very common portal of entry is our mucous membranes. So our eyes, our nose, our mouths, our ears as well, which is why when we had COVID-19 kind of protocols being put in place, we wore masks, we wore gloves in hospital settings. Some people had to wear glasses as well. That just prevents the virus from entering our mucous membranes. Bacterial infections like Salmonella, common portal of entry is through food, so our digestive tract. All right, moving on. Just because it has entered our body, it doesn't mean that virus or COVID-19 can actually spread within us. Okay? If we're immune to that disease already, then the virus is not going to be able to reproduce because our immune cells get rid of it. We need a susceptible host to finish off the chain of infection. Okay, what does susceptible host mean? It means someone who can be infected by that virus. They have not been affected before, they don't have immunity to that virus, and they have not had a vaccination against that virus. Okay, so in the case of COVID-19, someone who has not gotten the disease yet, or someone who has not been vaccinated from COVID-19, then the virus can reproduce within them and start off the chain of infection again. Okay, so I want you to remember each little part of the chain of infection. Tell me what it is, give me an example of what it includes. All right, moving on to the next stop point, I want you to know the three words endemic, epidemic, and pandemic. Okay, these three words go from order of magnitude. So endemic is the baseline level. Okay, um, every year, through every season, we have particular diseases that affect humans. For example, every year we get a few cases of the flu. Surprisingly, the plague, the Black Death, okay, we still have that in circulating in the world. It's not completely eradicated, and every year there are a few cases. But as long as those cases don't have a very big spike, for example, if the baseline every year we have 5 cases per year, we are expecting those 5 cases per year. They're not going to be eradicated. But if suddenly there is now 50 cases or 5,000 cases, then that is not normal. We call that an epidemic. So endemic is that baseline level that happens every year, the kind of the normal level. And epidemic is when there's a sudden increase, okay, within a localized area. This could include a community, population, or region. So if in Sydney there is suddenly a spike in cases that was previously 5 cases per year, now it's 5,000, we call that an epidemic. Pandemic, though, is when an epidemic has spread across multiple countries or continents. So, for example, COVID-19. Okay, that should be 19, not 10. COVID-19 started out as an epidemic in Wuhan because it was localized in Wuhan. But then very quickly it became a pandemic because it spread to all continents and many countries. Okay, so it slowly went to other parts of China and then it spread to many countries, and that's when it is announced as a pandemic. Okay, and the WHO or the WHO announces pandemics, and when a pandemic is announced, that's when all the countries have to kind of follow precautionary measures. So know these three words, be able to explain them, and don't get them confused, especially endemic and epidemics. All right, moving on to microbial testing, second in syllabus dot point. Microbial testing is one of the few examples of an experiment that you have to remember in year 12. You should have done it at school, so this is nothing new, and we have done it in our lessons. Okay, so microbial testing looks at testing for microbes in food or water. All right, so there are two types of experiments that you may have done at school. Testing for microbes in different types of food, so maybe swabbing cheese, swabbing bread, milk, and testing those, or looking at different microbes in different types of water. For example, tap water, pool water, pond water, and swabbing for that. I don't care which one you have done, as long as you remember one of them, because the HSC is looking for either food or water. The example that I'm going to give you here is water. Okay, you can remember whatever your teacher taught you. Okay, as long as you remember something. And I really want you to remember step by step what you did. Okay, this is one of the few experiments where it's just pure memory. Another thing that I want you to remember is controlled variables, independent variables, dependent variables, and the experimental control. That is something that a lot of students get confused about, the difference between the experimental control and a controlled variable. Okay, so let me just write this down. Okay, these two words often get confused, the experimental control and the controlled variable. Very quickly, in an experiment, we need an independent variable, the thing that you change. A dependent variable is a thing that changes depending on the independent variable. Okay, so in the case of this experiment, we're testing for different microbes in different types of water. The independent variable here is a type of water. Okay, so pool water, pond water, tap water, this is the thing that we are testing, the thing that we are changing in an experiment. The dependent variable is the thing that we are measuring, okay? The thing that changes depending on the independent variable. So for example, the dependent variable in here is the level of microbes or bacteria within each of these different types of waters. Every other variable needs to be controlled, otherwise it is not a valid experiment. Controlled variables can be the scientists testing it, okay? We don't want different scientists measuring the results, and this can skew our results. The environment that it is in, the temperature the agar plates are incubated in, those all have to be controlled. Without controlled variables, your experiment is not valid. Okay, those are controlled variables. Let's look at experimental controls. These experimental controls just test whether or not our experiment is valid or not. Okay, so if you don't have an experimental control, you can't really measure the validity of your experiment. So in the case of this experiment, let's read through the procedure very quickly. First of all, we say we prepare 8 nutrient agar plates in petri dishes. Then we label 2 as tap water, 2 as pond water, 2 as pool water, and 2 as the control. We then swab each type of water, put them across the agar plate, and label it with a sterile cotton swab. And we quickly replace the lid and seal tightly with sticky tape. Okay, for the control though, instead of swabbing the water source, we swab it with no water. Why would we do this? Okay, why would we put an agar plate with nothing on it, no water on it, and incubate that? Doesn't it seem kind of a waste of time? Not really, because an experimental control tests whether or not the only variable you are changing is an independent variable. Okay, so this agar plate for the control has a swab but no water on it. What this does is it allows us to test whether or not some other variable is changing. So, what we're expecting in this agar plate that has no water on it is we're expecting no microbial growth. Why? Because we salted with a sterile swab. There shouldn't be any microbes growing if the whole thing is sterile. So, if that agar plate does not grow any microbes, then we know we can trust our experiment. It is valid. Okay. However, if this agar plate, we incubate it properly, we sealed it properly, and it starts growing microbes, then what do? We can't do anything, right? Our experiment is invalid. So we have to chuck away our whole experiment and do it again. Okay, especially in uni, if you get invalid experiments, your whole results cannot count. Okay, it's not good scientific methodology. So if there is microbial growth, we can know that we didn't keep our controlled variables controlled. Maybe we got some pathogen infecting it. Maybe those pathogens in our air. Maybe we didn't seal it tightly enough. Maybe there was something else going on, and we can say that our experiment was not valid. Okay, so very important to know difference between these two, commonly tested in the HSC. All right, the rest of these read in your own time. I'm not going to go through it step by step with you. All right, moving on into Louis Pasteur and Robert Koch. These two scientists are very, very important for our understanding of infectious diseases. Right, they laid down the foundation for what we know today, especially the theory known as the germ theory of disease. So they didn't work together, but they worked upon each other's ideas to create and approve for this germ theory of a disease, which explains that microbes cause disease. What was before this? Have a little think. What was before the germ theory of disease? We had a few theories going on. One of them was the miasma theory. So people believed that diseases were spread through bad air, and they decided that it was the bad air or smelly air that caused diseases. A very common other theory was the theory of spontaneous generation, which looked at where people just thought that microbes or pathogens popped out of nothing. Okay, so they would think that if you left a bowl of ice cream on your table, if you left it for too long, microbes will spontaneously generate on that ice cream. Okay, so that's what they believed caused diseases. We know now that that's not the case. Okay, microbes cause diseases. If you leave ice cream on a bench for too long, then the bacteria within the ice cream will very quickly replicate and eventually cause disease if you leave it out there for like 2 or 3 days. Okay, so now we know that germs cause diseases. How did we come to this conclusion? We had Pasteur's experiment, his very famous swan neck flask experiment. Very quickly, what did he do? You should remember his methodology. However, it is quite long. So if you remember the general idea, I'll be happy with that. So what he did here was he got some swan necks. We call it a swan neck because the flask has this very curly neck, as you can see here, and he got a couple of these flasks. Okay, what he did was, okay, so this diagram here is a little bit simplified. It is a scientific model, and as you know, scientific models don't include everything. But what he did was he got one of the swan neck flasks. He put broth in it, so nutrient broth. I think it was like chicken broth or something like that. He put that broth in there, and then he heated that flask. So he boiled it to make sure that there were no microorganisms within that broth. All right. So he got this broth, heated it. Okay. He then got another swan neck flask. So he actually had a couple. This diagram here only shows one, but he got another one as well. He also heated that broth. But for the other one, he actually broke off the swan neck at this area here, this location here. Okay, so one flask he didn't break off the swan neck, the other flask he broke off. What he realized was after a couple weeks, the flask that he broke off the swan neck from, it started becoming really smelly. There was a lot of yucky microbes growing in it. So he realized that it was becoming really smelly, and he would definitely not drink that broth anymore because it was infested with microbes and pathogens. Okay, but very surprisingly, the other flask didn't have any microbial growth at all. Why was that the case? It was because it had this swan neck. And what happened was microbes in the air, came into the swan neck via gravity, but it all got clogged up here. Okay? Why? Because microbes can't just magically travel upwards, right? It's gravity pulling them down. So the microbes all got stuck here, and none of the microbes infected the actual broth. Okay, so what that means is microbes cause broth, and cells can only arise from pre-existing cells. That was his conclusion. Because his swan neck flask, he broke that off, he got microbe growth, that means it was microbes affecting the broth. Okay, so that's Louis Pasteur. Remember his experiment, remember his contribution to the germ theory of disease. All right, moving on to Robert Koch, also another one that you should remember off by heart and looking at what he did for the scientific community. Koch created these postulates, and postulates are things that are widely renowned as true in the scientific community. Robert Koch created these four postulates that allowed people to identify the particular microorganism which causes a particular disease. Because before Robert Koch, we knew microbes cause disease from Louis Pasteur's experiment. We knew that it could spread really quickly, but we couldn't actually identify which microbe was causing which disease. Why is this important? It's because we need to identify the thing that is causing the disease in order to prevent that organism to cause more disease. Okay, so in the case of COVID-19 again, we need to know that it's a SARS-CoV-2 virus causing COVID-19 in order to prevent that SARS-CoV-2 virus from spreading. Okay, so Robert Koch created these four postulates, which allows him to identify what microbe causes what disease. It is quite difficult, okay, to actually remember his postulates, but you will get the hang of it. I hope you do already remember it because exams are coming around. Okay, number one, the microorganism must be found in excess numbers in all diseased organisms and must be absent from healthy organisms. What this means is sick microorganisms, so sick rat, as you can see here, must have that microorganism within them, and healthy rat must not have that microorganism within them. Number two, the microorganism must be isolated from the diseased organism and grown into a pure culture. Okay, so we get this sick rat, we take out the microbe from them, and we grow it in pure culture. We culture it such that a lot of that microbe grows in that culture. Okay, number three, the cultured microorganism must be capable of reproducing the same disease in a healthy organism upon re-inoculation. Okay, what that means is we then get this grown microbe culture, we then swab it and infect a healthy organism. And what we should see is that healthy organism getting infected and diseased. Why? Because we just infected them with that all of that nasty microbes. All right, number four, the microorganism must be re-isolated and recultured from the inoculated and diseased organism. This cultured microorganism must be identical to the original microorganism. Okay, so we then get that rat that we inoculated with all the nasty microbes, and then we re-isolate the microbes from that rat, and hopefully under the microscope we can see the same microbe that we originally took out from the diseased rat. Okay, that is quite confusing, I know. Search up on YouTube. There's a lot of videos that really show this experiment being done, and hopefully your teacher has showed you how to do it. Okay, so this should sound very familiar to you. All right, number four, plant diseases in agriculture. This is a very small part of the syllabus, but I want you to remember one specific example, okay? If you are a little bit lazy, remember the example on this slide. You don't need to know more than one, okay? Panama disease is the one that I want you to remember. Very common example, comes up in a lot of trial questions and the HSC as well. So Panama disease is caused by a fungus, specifically the Fusarium oxysporum, and this affects the conductive tissues of Cavendish bananas. What are those tissues? They are the xylem and the phloem from year 11, okay? This causes the banana leaves to wilt and the stems to split. Why? Because we're affecting the xylem and phloem. Those are the systems that are necessary to provide enough nutrients and water to our leaves. Okay, and this causes banana leaves to wilt and unable to be sold to the market. And the reason why I want to remember this example is because there was a really big outbreak back in the 2000s And there was a period of time when there were no bananas on the market, okay, specifically Cavendish bananas. Right, that is the main market sold in bananas sold in supermarkets in Australia, and most of the Cavendish bananas are grown in Queensland. Okay, and what happened in Queensland back in the 2000s was this Panama disease rapidly spread through all of the banana farms. And because this fungus stays in the soil permanently, we can't really eradicate it. And it stays in soil. So soil is something that we can't just like dig up and throw away because soil is everywhere. So therefore, it was really hard to get rid of that disease and no bananas could be grown in that area anymore. So that caused a lot of impacts in the banana industry. And a lot of quarantine measures had to be taken place. The prices of bananas increased by a lot. I want you to remember this example, but if you want to remember another example, be my guest. Just remember the disease name, the pathogen that causes it, so whether it's a bacteria, virus, or a fungus, or whatever else it is, the name of the fungus, some symptoms that it causes, so wilting, discoloration, or some other symptoms, and then its effect on the agricultural industry. So, did it make the prices of that organism skyrocket? Did it mean that we had to get rid of whole banana plantations? Did it mean that we had to grow things elsewhere? Because it told me the effects on the agricultural industry as well. All right, another thing that I want to know is animal diseases in agriculture. Okay, so just like the plant one, I also want you to remember an animal disease. Mad cow disease is the one that everyone likes to remember. I love mad cow disease. It is really easy to remember and it is a one with a funny name. However, I want you to be careful with mad cow disease because it is not an Australian disease. Okay, mad cow disease never came to Australia. It affected European cows, it affected many cows around the world, but not Australian cows. What that means is if the question asks you for an Australian animal disease, you cannot put mad cow disease. Remember anything else? Equine influenza, foot and mouth disease, some other ones include bird flu. May remember any of those. Those are all Australian animal diseases. But if the question just says animal disease, mad cow disease is a really good one to remember. Okay, so again, similar to the plant disease, remember the disease name, remember the type of pathogen that causes it, so whether it's a prion, bacteria, virus, remember a couple symptoms that it causes. Okay, so mad cow disease affects the nervous system. This causes coordination being poor, aggressive behavior, and eventually death, and then some effects on the agricultural industry. So, did it mean that beef was banned from being exported? Did it mean that beef was really expensive? What did it have an impact on the agricultural industry? All right, so that is inquiry question one. Okay, in the next video, I'm going to go through inquiry question two, and that just looks at how animals or plants respond to infection. So I've gone through all of the pathogens, what they do, and how to classify it, some examples of those pathogens. Next time, we are going to look at the different responses to those pathogens. Okay, and just very quickly, if any of these concepts don't make sense, go back to my notes, read through them again, and then look through the relevant syllabus dot points. Okay. Another thing that I want to highlight is I do have an upcoming crash course coming up. So if you do want to sign up to that, we will be going through syllabus dot points in greater detail. We will also go through exam style questions. So it really puts this into application. Okay, registrations are going to be in the down below. So find that in the Google Forms and register if you are interested. Otherwise, you can also access this PowerPoint link down in the video and you can look at it in your own time. Otherwise, I'll see you in the next video.